The morning speakers, I didn't know if the music would stop and then we'd come on, or if we'd come on and the music would stop. So I think these two things are coming together. I'm happy to announce today we have a whole team of presenters, but Nasim has volunteered to go ahead and give the presentation for Dr. Kyle Elrot's Cancer Research Lab at OHSU, the Knight Center for Cancer Research. And we did want to give a content warning that this presentation will be discussing cancer research and the ways in which the team is engaging that. We also have two more of Nasim's lab mates. Quinn, you can show your bowling arm there. Quinn Wai Wong and Liam Beckman are also working in the lab. They're here to support Nasim, but Nasim will be giving the presentation. And when I asked Nasim, what would the audience need to know about you? And she said, well, I have degrees in computer science and in computational biology. And I turned my head sideways and I said, I know what biology is, and how do you compute that? And she said they build rocket ships. So in the presentation today, you should be able to see some Star Trek references and understand what the lab is working in cancer research. So Nasim, the floor is yours.
Thank you so much. Hi. Yeah. Good morning, everyone. I'm Nasim, and I'm truly excited to be here with my colleagues Quinn Wai, Liam, and our captain Kyle Elrot to discuss how we're building tools for genomics research at the Elrot Lab. We're part of the biomedical engineering department at the OHSU Knight Cancer Institute, which is right across Willamette River from OMSI and a hop away from Tillicum Bridge. Biomedical data is enormous, fragmented, and ever-evolving. At the Elrot Lab, we build scalable, interoperable tools that empower labs, researchers, and clinical trial tumor boards to unlock new insights. Before I dive in, I want to take a step back and highlight the fundamental question that drives our work. How can we find actionable insights in a disease as complex as cancer? Now, what do I mean by that? Cancer is incredibly heterogeneous. It doesn't just arise in one form or one tissue. It starts from normal cells, then it progresses to invasive disease, and it evolves in different ways across different organs over time. That means that no single data type can fully capture its complexity. Now, our mission is to build a system that pulls together a galaxy of data, clinical, genomics, transcriptomics, proteomics, imaging, into one cohesive system, where each of these modalities gives us a different snapshot, if you may, on how the cancer is evolving in that individual. Think of it like we're constructing our very own starship, where every data type or modality is a vital component. With this ship, we'll be able to navigate the vast cosmos of cancer data and uncover how cancer evolves in each patient. In the end, we want this platform to fetch and process actionable insights at light speed, so we can extract meaningful patterns in a timely manner for patients and to also enable groundbreaking research to iterate over what we have uncovered so far. There's a vast amount of biomedical data out there, but it's fragmented across different formats and standards. Enter our Captain Kyle's Enterprise D Starship project, an initiative built to unite this fragmented galaxy. In our project, data is the wrap core powering the journey, and our Python tooling is the glue that holds everything together. We use the Fast Healthcare Interoperability Resource, FHIR standard. That's the same standard trusted by Apple and Epic to pull and transform large consortium-level multimodal data into a unified graph schema. This unified schema makes phenotypic data points easily navigable in a graph. The FHIR Resources Python client leverages Pydantic under the hood, acting like a trusty droid to keep our schema components in check. Then with Python-based workflow engines like SnakeMake or orchestration platforms like Airflow, we're actively developing tooling for our ETL workflows. Think in-house bioinformatics analysis pipelines or modular graph builds. On the management side, we have built Grip, our graph integration platform that has a Python client called GripQL and serves as our command center for querying graph schemas. And alongside that, Gen 3 is the open source data commons and mesh we use to enable users to safely store and retrieve everything they need. There are global informatics standards our team has contributed to, such as genomic variant indices that speed up the search for gene or peptide base pair changes in massive VCF files, which can be as large as 3 billion base pairs or several gigabytes. We also use databases like Pathway Commons or Reactome that provide structured biological context and molecular interaction graphs. That's a graph of protein-protein interactions. This graph is pretty cool and is really where we link biological insights with data analysis findings. And I'll show you in a demo how we can use GRIP to find structured data, do some modeling, and then overlay our analysis features onto pathway graphs to fetch biological insights. We placed together some of these repos with hyperlinks. If you're interested, check them out, or come find us if you have any questions. Okay, let's walk through some code. Okay. Here I have already pulled the Cancer Genome Atlas, TCGA breast cancer data from Genomics Data Commons, transformed it with a Fireizer using Pydantic and Fire Resources Python client, and validated it with our Gen 3 tooling to check the nodes and relationships. So this is what a standard Fire graph looks like. It's pretty patient-centric. Then using this data and Fire schema both together with GRIP, I've created a graph called GDC BRCA, and I'm able to connect to it with GRIPQL connection object pointing to my local host and run a quick simple query on the patient node to check out the data, which looks good. Then I make a query to fetch this breast cancer's cohort molecular subtypes that has been annotated by experts in the field, and I'm gonna use these molecular subtypes to do a simple bioinformatics one-on-one machine learning modeling and build a prediction classifier for them. So out of this data, you can see a bunch of phenotypes, but the subtype, molecular subtypes are what we're interested in for our modeling. Okay. All right, so after loading and merging subtype labels with our normalized mRNA expression data, we perform exploratory data analysis just to get a feel of the data distribution. And you'll notice it's a bit unbalanced with luminal A having the most samples and HER2 the fewest. But when we reduce the data into a 2D UMAP space, clusters among them start to emerge. Those purple dots, for instance, you can clearly see the basal subtype patient samples, which are those purple dots here, standing apart. And this is pretty cool and makes sense biologically because basal subtypes don't rely on hormones like luminal A and luminal B do. All right, so we're going to choose a simple feed-forward neural net model. And with 2 hidden layers using ReLU activation, dropout for regularization, and a softmax output for multi-class classification. We split our data without replacement into 10 folds, performing cross-validation to check our model's performance and to ensure we're not overfitting. And we track 2 key metrics while we're doing this. One of them is accuracy, which measures the overall percentage of correct predictions, and the other one is F1 score, which balances precision and recall, a crucial measure when we're dealing with imbalanced classes. And the result of this cross-fold iteration looks pretty good. The average overall accuracy is 87%, and F1 score is around 73%. Then we're going to build one model over all of the data. And finally, to see which genes or features contribute the most to predicting each one of these subtypes, Since we use neural nets that have nonlinear activations in multiple layers, we're going to use Deep SHAP Explain, which is an enhanced version of Deep Lift algorithm and have proven the ability to fetch weights from neural nets. So for each subtype, we're going to choose the top 50 gene features and save them for our next step, which is pathway enrichment analysis. When it comes to pathway data, that's a graph of protein-protein interactions. I know it's a lot, and it looks like a pretty complicated and fun corn maze. But given my background, I like to make the analogy that pathways are similar to circuit boards in hardware, or in software, if you're familiar with color graph algorithms in compilers or state machines, then you can imagine that a sequence of protein-protein interactions converges to trigger cellular functions. And here's an example of how we can use this graph data and perform a pathway enrichment analysis. I can grab this pathway data from a community-curated open source resource called Reactome, make a pathway enrichment analysis API call that will overlay our genes onto the pathway genes. That's a contingency table problem to see which pathways are involved in each of these cancer subtypes. Here, I have chosen the basal subtype, and from a token I fetch from the API call, we can get a set of statistics and pathways that are involved in that subtype, generate a report that can support a patient's tumor board for discussions on care and treatment options, or even be published in open source paper to further our understanding of the mechanisms driving this cancer subtype. So that was just one example of the workflow that we can go through to catch some informative pattern, which we are working on integrating these type of workflows into our system. And every great starship needs an exceptional crew. Our team is led by Kyle Elrod and supported by our lead engineer, Brian Walsh. We have Quinn Wai, Liam, and Matthew as our software engineers, and Jordan and myself as computational biologists. And thank you so much for taking this journey with us. Feel free to scan the QR code for access to our code and slides. And I know that was a lot, so if any details I mentioned weren't familiar, please feel free to catch us after for a deeper discussion. Thank you.